The next part of the program is going to feature Dr. Gil Levin. And Dr. Levin is going to talk about the oxidant on Mars. And I'm going to turn the program over to you right now. That's fresh water.
Thank you, John. Well, I see I have to follow a very exuberant performance, which in turn was followed by the real McKay. I can only hope to inject a little Levin into it. And that's really what I've come to talk about. The Viking 1976 missions to Mars were billed by the National Academy of Science as perhaps the most important scientific experiment in history. That is, to search for life on another planet. I think it was very interesting that in the introductory films prepared by NASA, you did not hear the word life. What happened to that $1 billion search for life? Was the evidence so conclusively negative that it's not worth talking about anymore? Or was there some reason to denigrate the principal purpose of the mission to, as it appears in the new book, Mars, a book of some 1,500 pages, to confine the chapter on the search for life to 11 pages. And in that text, to merely say, there are some scientists who believe that Viking detected life on Mars, but not to present their case. The truth is that for all of its promise, and despite the fact that it was probably the most complex space mission of our time, Viking did not solve the riddle and leaves us, as its legacy, two very important scientific problems. Did Viking discover life or some oxidant on the surface of Mars is the key problem. But another problem, really equally as important, is where are the organic compounds that everyone knew was going to be on Mars and constituted the reason to send a very sophisticated machine to determine which organic compounds were there. Not whether there were organic compounds there, we all knew there were, but which organic compounds were there. Strangely enough, the two questions remain. The Viking carried on each of its two spacecraft the contingent of 3 experiments to look for life, plus an experiment to identify the organic matter in the soil. I was experimenter on one of those experiments I would like to talk about, the so-called labeled release experiment. Originally, I called it Gulliver, but that didn't fly well with the bureaucrats. They did not see the analogy of going to a strange place to look for strange organisms, so they gave it the much more erudite name of the labeled release experiment, which we've contracted to the LR experiment. The rationale of the LR experiment was we could use the very, very sensitive method of radioisotopes to look for life. And the life we're looking for are not the green-eyed monsters, but tiny single-celled microorganisms, which we felt that were there any life on Mars, there would have to be microorganisms, because even if there were elephants, which would probably be hard to catch, they would require microorganisms to decompose them and keep the life cycle going. And if microorganisms on Mars had pursued a path of those on Earth, they would be distributed as a thin film throughout the entire surface of the planet. So this was the place to start looking for life. And we selected the most sensitive tool that we could. It was a tool that supplied radioactive carbon compounds to portions of soil, and were there anything alive in that soil that could utilize the organic compounds hopefully, these organisms would digest the food and, as all organisms on Earth do, produce gas. The gas being produced would now be radioactive because the food was radioactive. And so we concocted a food drink made of very simple organic compounds labeled with carbon-14. Now, we didn't just go to the grocery store and pull these organic compounds off the shelf. We used two approaches. One was theoretical and the other was empirical. On the theoretical basis, we looked for materials that had been formed in experiments such as the Miller-Urey experiment, which produced amino acids, carbohydrates, and other building blocks of life, such that it was presumed that early Earth, which had a primitive atmosphere, which was the atmosphere that Miller and Urey exposed to ultraviolet light or electric spark and produced these organic compounds, would have been the method by which life got its start, from the compounds produced abiotically, which in turn would somehow be complexed into the building blocks of life. So we used only compounds that had been formed by Miller-Urey type reactions. But secondly, we tested those compounds on terrestrial organisms. Just as Chris, we roved the planet and went to all of those exotic places or received samples from them, tested thousands of soil samples, thousands of unicellular cultures, pure cultures, wild cultures, against these compounds. And in every case, received responses to tell us there was something alive. If I can have the first view graph, please. Oh, that tells us that we're here. Next view graph. This is how the instrument worked. You saw the arm that went out. It put some soil into a distributor, and about half a cc of soil was plopped into one of 4 rotating chambers. A tiny squirt of nutrient was injected onto the soil. Were anything alive or anything generating radioactive gas, the gas would come up, go through this swan tube, the same kind of tube Pasteur used, that's his link with Mars, and go into a chamber where the radioactivity could be detected. The radioactivity would be monitored through time as it evolved from the chamber. And the beauty of this was that you would use a known amount of radioactivity but never have to worry about counting that or subtracting that from your background because there was a phase change. The radioactive material would have to leave the chamber where it was deposited, go through the Swan's tube where any particulates would be removed and only gas would come through, so you don't have to worry about the background of the nutrient that you originally injected. After all of the experiments were done on Earth and the system was bolted up and Viking was launched to Mars, at that time we did our final test. It may sound a little in arrears, but the schedule demanded it. This was the only time we had a full Viking labeled release flight type instrument. It was taken to Ames Research Center, put into a Mars tank, and some California soil was put into the labeled release chamber. The whole experiment and the chamber was then pumped down to Mars conditions, to Mars experimental conditions. The Mars atmosphere, the temperature 10 degrees C at which this instrument would be maintained through its experiment. This pumping went on for 3 days, and then the experiment was initiated, and the following resulted.